Okay, good morning. So today is lecture number 4. For this week, we are going to discuss probability and distributions. As I said, machine learning is stayed on 2 pillars. One is lineage algebra, another is statistics. So statistics modeling and inference will be very important part of this to support machine learning. You can add more pillars like optimization or other, but those are major ones. Last week, your homework is about lineage algebra, how to do the matrix multiplication. So homework has been done and very well. And today we are going to have another homework which help you to understand this cosine distance and that formula from previous lecture. Okay. So today we are going to talk about the concept of the general statistical modeling. And you heard more about this generative model. And so this is what we call to have the model of the statistics, and from there to pop up those numbers to fit the expected distribution. So there are some special case in doing the statistics in single cell data, because we are dealing with the counts, and also is a non-negative. There are zeros, 1, 2, 3. So like normal distribution, we expect to have this continuous variable to model. Well, for these counts, these integers, we use Poisson distribution, negative binomial. So I will mention about those. Then we will have very general coverage for the mean and the variance. I hope you all know this before, but for the systemically go over this. So I will still show some box plots and cumulative curve, etc., etc. Then we will focus on how to calculate covariance matrix. All right. And at the end, we will mention the KL divergence. Whoever, which field you are in, if you work in the machine learning, if you want to compare two distributions, people use KL divergence a lot. All right. So along those, we have some other small terminologies, small concepts you have to know. So we will cover quite a lot, but it's a very general overview. All right. I mentioned this in the last week computer session. So if we have a count matrix looks like this. This is a real count matrix, UMIs. And you will see some genes highly expressed. In this case, is ISG 15, right? Some genes are lowly expressed. You can see its expression level in the CDK11A, right? So this is a real case. So one cell type looks like this. If you change the cell type, you may have a different profile. Okay. So what we can do with this table? If you have this table, in general, we don't deal with this in Excel spreadsheet, but you can, if you like, if you have a relatively small matrix, you can put it in there. So what we can do is we take average or compute the summation of the column, the total counts for every cell. So if you sum everything together, so what's this? So this is library size, we call basically how many rates have been mapped in this gene, in all the genes in this cell. So if you sum this together, you get a number. You sum this together, you get another number, right, by column. If you do this by the rows, that will be the average or total expression of genes in those cells, all right? So I transpose this matrix, so just make this display easier. So I put columns as a cell, and then genes, it's still the same. We are going to compute library size and then do the normalization for the library size. So this normalization is not unique for the single cell data. Library size normalization has been used for bulk RNA-seq before, all right? So bulk RNA-seq, you have columns, not individual cells, but the samples. Normally, we have, for each group, you have 1, 2, 3 biological replicates. You can have 3 columns. That's your 3 samples. Then in your other conditions, you may have another 3, or people say, okay, 3 is not enough. You have 4 or 7 in order to do the differential expression. Same idea. So here we borrow the library size. We inherited the terminology from the bulk RNA-seq. We still call it library size, but it's basically how deep you sequence for each cell. All right? So if there's no technical bias, we expect all the cells should be sequenced to the same depth. So largely, you should have a similar library size. So if you summarize this column, and it should have a similar number to the next one. So we expect all the cells in this population in the same technology, same library sequencing batch, you should have a similar library size. That's an assumption. We will see it's not true because some cells, macrophages, are larger than T cells, so you expect to have more RNA in there, okay? But we are talking about the library size normalization in order to control for technical bias in sequence depths. This unavoidable, is inevitable. So you always have some variability. If you sequence even the same cell, again, you have the same, you don't expect to have exactly the same library size. So this is by gene expression, this is by column. So you just put this, I use a small example to show. And this cell has been sequenced more in 10, and this cell, unfortunately, only sequenced twice. So that's why we normalize it, okay? If you have 7, this every number here will divide by 7, okay? If you have 10, every number in this column will divide by 10. So after this normalization, if you sum the column, you will get 1 for all the rows. So you normalize the library size. Because if you sequence this deeper, I will divide by the total and give you. So that's what this happened, okay? But instead of put 1, we will get the medium, so we multiplicate this 1 by a factor. Okay, in this case, 5.29, because that's the medium library size. You can scale this to any numbers. If you like 1,000, you can say, okay, after I divide by the total library size, and I multiplicate by 1,000, then your library size will be uniformly across all the cells, become 1,000. That's okay. Okay, you just scale everything to a factor. So this is called library size normalization. This has been used in bulk RNA-seq all the time. You have to do this before you do the comparison across samples. So we are dealing with relationship between cells, so we need to do this normalization. So this is MATLAB code. So you do some function to get the library size. This become vector because the sum by default, this function will calculate the total by columns. And then divide by library size. So you have a whole matrix, and every matrix, every column will divide by the corresponding library size for that column. So this operation, put a dot, it's element based. So it's, this computer language is smart enough. They will help you to divide each column by its total. And then you get a normalized version. All right? Then you multiply it by an arbitrary number to scale it up. In this case, 10,000. Then you get a normalized version of X. So the procedure I just described is this. So you have row matrix, you have genes, and you have samples in columns. In this case, is a bulk RNA-seq. You have patient and control. It looks like patient has been sequenced as much as twice as a control group. Okay, there's a technical bias. And then you go this way to do the library size normalization. And you turn this down to 60, okay, by a factor. Now, patient and control have the same number of the total library size, and then they make them comparable. So you can start to compare this gene against between two different samples. In bulk RNA-seq, we also consider how long a gene is. So the gene body in gene 1 is 5K, while the gene 2 is only 1K. So by itself, you should expect have twice number of rates mapped to the gene 1 because the gene is longer. So you have more codons or exons in there. So this is called RPKM. So you controlled for the library size and also consider the gene body length. We are not doing this in single-cell RNA-seq because single-cell RNA-seq does not care how long the gene is, only capture the poly A tail. So you only get information from the part of this, the end of poly A tails. All right. So we don't do this RPKM, so which is standard library size plus gene body length normalization in bulk RNA-seq. And in the DE-seq, the most commonly used differential expression analysis, they're doing this slightly different ways to do the normalization. So for us, we just remember this, okay? So normally we will do this, but there's not, as I said, there's some problem associated with this. This normalization is very, very rough. We don't consider the different cell type have a different library size expectation. As I said, the macrophage is larger than lymphocytes. So you expect library size should be by itself larger than the library size you sequence from lymphocytes. So neutrophil maybe have a similar or in between this, okay? But we cannot consider this because of we have no idea what the library size you sequenced from one cell is because by chance you sequence it more, or by chance it's a macrophage, it's larger by itself. So if we don't consider, we just do the normalization as a usual way, this will not fair to macrophages because you'll force the normalization, force it to push the average library size to the same of all the three. Correspondingly, all the genes in macrophages will also be, expression level will be also pushed down. So even though this housekeeping gene is supposed to have the same numbers expression across all the, because of the library size normalization, you make this even slightly biased once you, when you estimate the expression of some of the genes, okay? Makes sense, right? So that's why we want to have some more sophisticated ways. Okay, just assume library size are the difference because of a technique of sequencing bias causes this variability across cells. And also, there's cell type difference. Okay, sometimes you have a high library size because the cell is bigger. So we want to do this very sophisticatedly, and you can have assumptions, say, okay, this is macrophage, and this is a T cell. And then you do the normalization and have an expectation of library size, do the library size normalization, put the cells in different categories. Then you come back and do the estimation again, see after normalization whether it is true for my assumption because I assigned some of these cells to macrophage group. But they are probably is because of a technical bias to push the library size artificially high. So they're not true. So there are some statistics you can use to do this iteration. Okay, try to fit 3 categories. You don't know how many categories in the cell you sequenced. Okay. So, so this is one of the modeling procedure people use can solve this problem. However, this biscuit, the library correction method, and also you can do imputation and clustering at the same time, is not widely used, although it's statistically very sophisticated. The reason is they are slow, so you have to have assumption to have hyperparameters to set how many categories and change. If you need one more category, you go through this so-called Chinese restaurant process to add more tables, more categories to allow people to sit around. And if not enough, you set another table. So you don't see the application in real life because people just cannot wait. I want to normalize my data and I want to move to another step. So we don't use this at all nowadays. So we still use this because this takes no time. It's done, so normalized, and move towards next step. All right, but you need to have some idea. Okay, this is a problem. So now move to statistical modeling. So when biology say modeling, and you probably say, okay, I work on the system in animal or cells. And at the end, I put a model, so this gene have a regulation relationship with that gene. This gene will inhibit another gene. So you are building a model. When people in statistics department say modeling, they mean they want to look at the data and think about what the data distribution looks like. Okay. So that's the modeling step. You have the data, you have events, you have a collected histogram of your data points, and you want to do statistics to model to build a standard distribution so you understand how much you expect when the value should be located in this bin and rather than this bin. So you do this and to understand, so this is the model basically, the distribution, and you have a theta is a parameter, and you want to generate your data from the model. And this model should fit your expectation very well. So this is a procedure, it's called statistical modeling. So I'm going to use real examples to illustrate my points, but the most important is those days should be generative model because people use a lot to generate the next word in the sentence should be. So before that, we need to have an idea about what's the distribution, what is the modeling about. So my example is like if we are in a task, say I want you to generate a list of numbers to tell the height of the man in from Italy versus Sweden. So you may have idea, okay, Sweden taller in general. So if you put a number in this column, if by centimeters, I probably will put the first Sweden people, so Italian. In this case, I may put 180, okay, centimeter. I just simulate a survey on the street, okay? And in this case, I may say, okay, Italian is relatively short, okay, in my idea. Where I get this idea, because I heard the news or from whatever information I have, that's your assumption that's your model in your mind. So this is a statistical model. So I'm pop up number, so I make this 174. Okay, another here I will see, okay, 970. Where here I put 167, 70. Maybe they're several higher than that, but I know if I put it longer enough. Is that the case? Okay. Because I have, in my mind, I have modeling. This model is Sweden. My model is normal distribution model. So this distribution is for Sweden here, well, for Italian here. So you have a different mean. So I know I should regenerate this distribution using some synthesized data, but I need to have the idea where the mean is. So I have a difference between them. So on your mind is doing a generative model to simulate the real, okay? So in our case, we will simulate a gene's expression across cells. So you need to have some idea about what this looks like. If you want to simulate, this is real data, as I said, this is real data. So you need to have some idea about ISG 15, what is average. If this average, you think, okay, this gene should be here, it should be a Swedish, and then some other genes like this could be Italian, okay? But in this case, we are not so sure these numbers follow the normal distribution, because the normal distribution is not the real discrete distribution. It's not the best model for simulate this. This is good for our understanding of the heights of the people, because height is a complex trait, and it's encoded by genes or environmental factors. And in terms of gene, there are thousands of loci in your genome determine how high you will be. So that's our problem. How we model these numbers and assign each theoretical expectation for each gene. That's our problem, okay? Come back to here. Okay, so this procedure will be, we look at the numbers. We do a statistics, we count. This 0, and you have several cells for that particular gene, count as 0. And when you have 1, this number of cells. And then if you have 2, then you have more cells in 2 group. If you do this, you get a histogram, then you do histogram, then you, from here, you infer as a continuous distribution. This is like a theoretical solution for the best fit for this. You make a curve in order to allow all the counts fit this shape. And with this expectation, if you generate the numbers it will generate more from have an average in this for the gene expression across cells, and generate less cells with this gene expression at this level. So this is from data. You look at the data and try to figure out the distribution. Okay? So for this case, it's relatively easy because from a large data statistic, we know this trait, the phenotype, fit normal distribution, Gaussian distribution are real. So we can get enough data points and fit the data and have a mean, have a variance. These two numbers will decide the shape and the location of the peak of this curve. And that's the modeling. That's from data to distribution. So once you have this distribution, you can generate this data back and very accurately. All right? Well, our question is for the real data in single cell, even after normalization. Okay, so which model we should use? There are many, many models in statistics, many, many different distributions. Statistics found negative binomial distribution will be the good one. So this is good for the count numbers. Okay. So if you assume, for example, this is the distribution looks like the mean and the variance. If you assume the mean is 7 and the variance is 0.3, and then you will have a distribution histogram looks like this purple, this light blue curve. Well, if you have average, see, okay, it's negative binomial. If it's 1 and 0.2, it will look like this. So what you can do is, if you have a large number of cells for particular gene, if you have a gene histogram looks like this dark red, and then you can see, okay, the negative binomial distribution parameters for this gene in the cell should be 1 and 0.2. And 1 is the number we want. We want to see average expression for that particular gene in the system if we assume the distribution is negative binomial. Well, for another gene, if it is yellow, so you can see, okay, it will be 10, okay, with fitting the variance. We will have to look at the shape of this as well. So why we can do this? Let's look at a function. So in MATLAB function, it's called negative binomial random number generator, okay? NBIN, negative random. So we know if you do R, A, and D, and you can generate a random number, but that number is from the uniformly distributed number. So it's from 0 to 1, and this could be 0.5, 0.56, 0.7. So that's in the middle of from 0 to 1. But this function is also generate a random number, okay, is a count, is integer, will be follow the negative binomial distribution. You need to put two numbers in this in order to generate one random number. Okay, this number will be an integer. So what's those two numbers? You need to have R. R is probability of success in a single try. Let's see, I shoot a basketball to the basket. my success number is if I 0.3. Okay, if I shoot 10 times, I may have 3 times in the basket. So this is like a probability you can get on average. If you single try, then you have R. And then another is to the probability is P, so that my success rate to shoot the basket in basketball in the basket is P, okay, 0.3. And this is the number of the success R. So if I have see, random corresponding number of success. Yeah. So let's do this as an example. So this example is simulate defect probability. So you produce something, and then in your series of products, the defect rate is 0.01. So you produce something, and then you found one could be have some problem with defect probability. In 100 you have 1, so there's a rate. So your question is how many units might your quality assurance inspect before found 3 defective items. So how many you need to check before you found 3 defective items from the product? So if you do this, you put 3 here, so that means you found 3 defect products. If the rate is 0.01, so you need to try a lot in order to get 3 defect ones, right? So this is what we will generate. That's a P. Okay, you have 3 defects. That's a P. That's R. So P. In my basket shooting is how many shoot I can get 3 within the success. How many shoot I need to try before I can get 3 success. So that's basically we will generate the number there. This function here is to let this program to run 6 times. Okay, so sometimes you need 496 trials before you can get 3 defect ones. And sometimes you only try 142, you get it. But the average number will follow that distribution. But because it's random generators, so you don't expect the same number, all right? So add 3 means because you have 3 defective ones, so you add 3 in order to total number of trials or detect events. So you need to check, in this case, 496 products in order to get 3 defective ones. Remember, if this number is small, if you have 0.001, okay, 10 times small of the defect rate, then you need to try more in order to get 3 defect ones, right? So we can test this in the real computation. But the idea is you have a small probability event, in this case 100%, 1% defect rate, and then you want to get 3 defective items, how many tests, how many products you should check in order to get this. So why we need to use this? So let's change this language. See, in the single cell data, our technique have a detection rate, detecting gene expression successfully in 1%. If you have a gene expressed in that cell, my technique will detect it in 1%. Okay, so very low rate. then we will see, okay, how many mRNAs that gene has to be in there, has to be allow me to try in order to get the UMI count to be 3. So you need a larger number of abundance of mRNAs because my detection rate is very low, only 0.01. But once you have abundant mRNAs, give my higher chance, basically you have increased number of possibility of trials. And then when I read out this UMI in this cell is 3, I can infer back what's the original numbers of mRNAs in the cell. You have tried to think about get your head around this. So we detect the defect product here, but now we are detecting UMIs from the cell. We detect 3 as UMI of that particular gene's expression, but that gene should be, expression level should be 496 or 142. They're a much larger number, it's not 3, because our detecting probability is low. Okay? So this is a use of this modeling to see, okay, to estimate the real expression of the gene. So how many copies of mRNAs should be in that cell if we detect UMI X3. So that's indeed a lot of this single-cell RNA-seq estimation is based on when you observed UMI equals 3, so there are some assumption of the detecting rate and some others. So you have your capture rate and the dilution factors, etc. You can model everything in the middle of the process you believe will reduce the detecting rate. And you build everything into your model. You also consider GC content and transcription length. So gene may have different GC content and will bias the detection. You can put all the amplification, capture, and the dilution, everything. You come up with a good estimate of real mRNA concentration in the cell if you observed a number of UMI. So the procedure can be very complicated. But the idea is to use this probability. If you look at those, people will do normalization, and also sometimes they try to estimate the empty values. And they believe some of the genes, if you have 2 here, 10 here, you don't expect 0 because these are the technique drop out. Maybe we should do the imputation. So they want to model the negative binomial distribution. I try to fill in those values. So this procedure is called imputation. Basically, we'll be trading those zeros as missing values, and then try to use their understanding of the statistic distribution to fill in the values here. So 0 could become 1 or 2, depends on what your model is about. There are a group of the model-based method to do the imputation. If you look at their description, a lot of them will mention the negative binomial distribution because they believe there's a small event happened with the capture rate is low. So you expect higher numbers of the real expression count, but you only detect the UMI small numbers. So that they want to use this to simulate or push back, fill back the values in the sparse matrix, make this more dense expression. There are other data driving and the smoothing technology. You treat this use the neighbor values to correct the missing values. There are also, so there's like a neural network based, and there are some others. So statisticians have been trying to understand the distribution to do the imputation, okay? So they believe if you impute this correctly, you should be able to enhance the downstream analysis, because this sparse matrix become dense matrix, then you can do this distance computation easier, and those will help you to do the clustering, to do the differential expression, to infer the relationship between cells, and to help you achieve better results. But everything here is based on your understanding of the system, and there's nothing wrong with this. But in reality, it's not the case, so we don't recommend to do any imputation in the real data analysis. Okay. So Saver is one of these model-based algorithms. So we open it up and to look at how this has been formulated. So the Y is a gene expression level in the cell. So G, C are index for the gene and the cell. So if you consider this as the UMI count for the gene 1 in the cell 1, it's a Y equals 3. So this number is a 3. The model assumes this number of the generator from a Poisson distribution. So you have a lambda, which is average expression. Well, this average expression is not the same across different genes. So gene 1 may have a higher, gene 2 may have a lower. So in order to describe this average gene expression difference, they use so-called gamma distribution to generate a very variable shape and to fit the gene expression difference from one gene to another. So once you have this modeled, and then you put that in to give the Poisson distribution, and it will generate. So here they use Poisson instead of a negative binomial. You can change the Poisson to a negative binomial, and then to achieve the same thing. This is another one. So this is called SC impute, another pipeline to do the imputation in 2018. Let's see SC impute from which category. SC impute is also model-based, one of the methods in here. Well, to do normalization, detect subpopulation of outliers, and then identify the dropout values in the mixed model, and then negative, non-negative least squared regression neighborhood. Yeah, so this is the idea is to do the dropout values and to put back values to non-zero values. So I think what I want you to get familiar with this is this equation. So once you see this double bar, that means what? So this is a norm. So if you have say V, so that's a norm for a vector. So this is the magnitude, how long this is. The formula in here, so this one tells you, this is called, when you put a 2 here, this is called L2 norm. Then the squared. So they basically square the difference between this and that. So they try to find the values of beta, which some parameters, and help to make these two matrices value as small as possible. So you want to find, when you have an argument, so you found the parameter combinations to make the whole norm as small as possible. So this is a formula basically people will use to let this to approximate this as much as possible. So you will see quite a lot if you do the optimization. The purpose is to make these two matrices. X is your observed, and this is your modeled as much as possible. Okay, so let's see, people, so this you're all very familiar with, and you have descriptive statistics. We want to have the mean. You can use median and the mode to describe the central tendency of your distribution. Okay, you need single values to describe this, and also you want to have the most important the dispersion and the variance. So variation. So basically you use variance, standard deviation, or range. So that's what you need to remember. Then there do the inference, including statistic test or estimation of parameters. Okay, so the parameters and also confidence interval. So this is what a statistician is doing normally. So terminology, PDF. PDF is a histogram or frequency, how to say, histogram about your values, okay? In this case, it tells you the frequency of, in this value, in this, and the frequency which your sample will located in this value will be lower and there, okay? There's some normalization procedure to make though the shape and here become frequency rather than the real count. So this is called a PDF. You have this. And then you can put all the bins onto each other. So you build this so-called CDF, the cumulative distribution function. Rather than to show this peak, you change this by adding everything together, and you get this cumulative curve. This is from 0 to 1. Okay, your curve, you will eventually reach to 1 once you give all the possible values for this distribution allowed range. And this is called box plot, and this is a horizontal way to show, or you can put a vertical. And in general, in the middle, you have this bar indicated the median. And then you have 25 to 75 quantile, and then that means there are only 25% of points lower in this bar, so mostly in this range. Should be have a 50% of data should be in that range inside the box. And then you can define a cutoff. So anything above this range will be outliers. So those will be outliers, and this will be outliers. So box plot is very commonly used graph presentation of your distribution. So this gives you a lot of information about the centrality trend of your data distribution and also dispersion information. So this is PDF, CDF, and the box plot put together, and we can see if you have a change of your distribution, what will the shape will change. So this is a very normal distributed data distribution. Then you have a CDF looks like this, and very standard symmetric, okay, box plot. You have outliers in both sides. Well, if you have a shape of PDF look like they're plateau in the center and very wide, then you have a slow increasement in the cumulative distribution. This shape will become less steep, and then you have a larger range in the middle compared to this normal, right? So this range is here. Well, this becomes a plateau here, and then your box plot have a larger range in order to contain 50% of those data points in this range. Well, in contrast, if you have a PDF with a sharp peak, and your CDF will have a quick increase in the middle with small range of exchange, you will have a quick increase of the cumulative probabilities in this range, where your box plot will be very narrow in this box. If you have this skewed distribution, then your peak which skewed to one end, and your box plot will look like this, okay? This is probably most likely to be our single cell data because we have a lot of zeros or small numbers. We have this bottom cutoff. You cannot go negative values. So you may have some cells for that particular gene's expression really high. So this will be box plot we will see a lot if you do a real untransformed UMI count plot for some genes from real data set, from single cell. And if you have a PDF with bimodal, that means you have two peaks, okay? You may be able to capture this in this change of the slope in the cumulative curve, but you will lose that information completely from a box plot, okay? Sometimes that's why you need to plot both box plot and PDF together in the same figure in order to have a good representation of your distribution of the data. This could be true for some genes in your population of cell. If they have two states of your population, one group of cell express that gene in the low level, that will be here, okay? Will another population of cells express that gene high in this level. So you may see these two peaks. It could be interesting genes, we don't know. All right, just some terminologies in here. I think when you do the statistics, you want to see whether your observation is located in certain cutoff. This will be 95% significant because you don't expect your result will be here if you estimation on average should be in the middle, because this level is really low. In total, you only count for 5% of chance you'll be in that region or above. So that's where the statistics is about. So if you have a normal distributed data, you can easily detect. So this could be 181. This could be outliers for the Italian, because you expect very few cases you have this, but for Swedish, it may be just located under the curve in the middle. It's hard to do statistic inference or this test for single cell data because we have no idea what the distribution looks like. But at least we know most of genes are not distributed in a Gaussian distribution. So you cannot just compare one gene in this group of cell to that group of cell by using the t-test. It will still work, but not accurate because your distribution is not follow this beautiful curve. So you need something else to do this. Terminologies. So people use normalization, sometimes use standardized. They mean different things. And when we talk about the standardization, so we will see, okay, there's a z-score transformation. So you have your values minus its a mean. So you have a lot of a vector of data, and then you minus its mean and divide by the standard deviation. This becomes a z-score. And this score is good because you don't worry about. So after you normalize, you will start be able to compute a normalized z-score across different standardized. Okay. So this is a transformation, kind of transformation. I think I will quickly explain this multiple testing correction then before we take a break. So who have you used multiple test correction before? Okay, Sarah, yeah. So the idea is if you observed, so you do the test, so you do a lot of tests, you have p-values. If you are doing just one test, if you get a 0.01, that means what you can observe that event happened is only 1% by chance. So this looks like very significant. But if you repeat the same test for, say, 10,000 times, then if you have a uncorrected p-value 0.01, so this will be not very significant. The reason is in so many tests, we are basically recreate the whole distribution like this. So there's some chance you will, for sure, will get 0.01. For instance, if you are doing this bin machine, okay, sometimes you found this in the museum, and you put a bin in there. So in the morning, for example, you come and put one bin in, and this bin jump to here. So you will find that it's very strange. It's a significant event for you because you drop one bin, and this bin this morning come to the one of the end of here. So that's an event which you have uncorrected test, but you get a 0.01 p-values p-value by just one test. However, if you keep drawing, if you didn't get that, but you keep drawing this the whole morning, you put one bin, and after, in one of these tests, you put 1,000 bins here, and there's one drop to here. Okay, in that case, this event is not very significant because when you do this 1,000 times, you expect one of this or two of this should be to the very end, because you are repeat your same experiment again and again. You are rebuild the whole distribution, so you expect some of these bins should be in there. It's very different from the in the morning I just described. You come in the museum and drop one bin, and this bin jump to the end. This very special event. Okay, so that's why in order to claim this drop of the bin and the bin come to the end, you need to take account how many tests you did. Okay. Now, in this case, if it's only one time you drop this and come to the end, one side of the bin, that will be significant events. Otherwise, you repeat, it's not very significant. So people use multiple testing corrections. Just normally, you just divide by your observed observed p-value by basically you times this, okay? 0.01 times how many tests you did and make a Q value. Q value means you are correct for the multiple testing. And then you multiply it by how many times this becomes very insignificant. So how can you achieve significant after correction of multiple tests? You need a very small p-value, say 0.000000001. Then even you multiply it by 10,000, you still have a very significant Q value. Okay, so that's the correction for the multiple testing. This is essential if you have, in the genomics or the transcriptome analysis, you have so many genes, you test one gene against another again and again. You basically repeat the same test many, many times. So you need a very stringent cutoff. This cutoff will be, depends on how many tests you get involved in your trials, all right? So that's a Stokes-Roney correction. So if you have say 100,000 times tests, you need to have p-value which at this level in order to give you the correct version of the Q value, the correct p-value 0.05 significance. Okay, so 5 minutes break, come back 11:07. Okay. Okay, so everyone is back. So, so this chart illustrates the link between different distributions. So we put normal distribution in the center of this figure, and then with some change of parameter and restrictions, it become t-test, student t distribution, and then negative binomial is somehow linked with geometric distribution, and then it's linked with a binomial distribution with a Poisson and a special case, and the Poisson becomes a normal distribution exponential. So everything seems linked. So we are going to quickly mention Poisson distributions. So this is also for the some numbers of success in intervals of independent. So we have a probability of success. So you have average basically. So people use this to describe this event number of happened. So for example, this is how many. So if you know the average of the car pass a street is like per hour, you have on average your statistic is 5 cars or something like that, and then you will estimate in certain period, so every 5 minutes, and how many cars will pass this intersection. So you have this distribution looks like this. If you have a lambda equals 3, then you put into the PDF function for Poisson distribution, then your average is lambda is fixed. So your average, your long-term average is 3. Then how many, what's the probability if you have 1 car passed, if you know the average is 3? So the probability is 15%. You can observe. You can observe if this one car pass. On average is 3. So what's the two? The two probabilities something above 22%, and the 3 you will get this. Then it's very unlikely you have 8 or 9 car pass in the same interval of the time. So this is the distribution of a Poisson. You only need one parameter, which is this average, in order to reconstruct the distribution. This figure is average is 3, so you get a curve like this. Well, if you have average is 4, you get a curve this dark purple. If it's 1, you get the Poisson distribution looks like this. If it's 10, then it becomes close to the normal distribution, because this is a large average. So in the single cell study, we used quite a lot to describe this different stage. We can use Poisson distribution. We can set the average rate for the cell into the droplet, okay, the small event, and that's the percentage, the likelihood, the probability of this, okay. We can also describe the rate using mu here, and then this is like a procedure. You load the bee into the droplet. So this all technique steps if you want to simulate or model every single steps, you can have this small event model to simulate those. So people also use some addition features. So any model you can add zero inflated. Basically, you want to model the zeros as a separate category. So you see, okay, I'm supposed to express, observe 50% of zeros. So you set values to allow you to put the data into two categories. One use the model, another use this estimate zero inflated rate. So you may see a zero inflated Poisson distribution model. Basically, you treat zeros very different from others. So this is a comparison between negative binomial distribution, and you see a lot of this here. There's all zero. Then zero inflated. So you receive different PDF if you consider zeros as a secondary category and model this inflation. The last distribution I want to mention is the T distribution. So if you look at the chart I just showed you, T distribution is linked very closely with normal distribution. Okay, where's my? Yeah. So that's where they linked. The shape is very similar. So if I don't tell you, you may not be able to tell which one is T distribution, which one is normal distribution. What's the difference is this. So T distribution have a what we call its fat tail, okay? So if you consider this as a tail of the lizard or dinosaur, so this tail is so if you look at the normal distribution, they have a very skinny tail at the end, but the T distribution have a fatter, okay, tail. So there's a difference between them. Why I'm going to mention this, because this feature we are going to use in the maybe next. So we have a TSNE, dimensionality reduction technique. So in this technique, the TSNE technique, T is a T distribution. So this technique utilizes the feature of T distribution, have a fatter tail to learn the distribution using normal distribution, but regenerate data using T distribution. Okay, so you learn your centrality of your data using normal distribution, and then model it, use this normal distribution. So there's some of the expectation in here. So you expect this level of data generated from close to -3, but when you regenerate your data distribution, you put a lot of weight in here. Basically like the Italian and the Sweden people, so you don't expect to have Italian have 190 centimeter, but in reality, when you simulate it, you put a lot of 190 in that end. So make Italian people, the distribution more, how to say, close to T distribution. So we are, in this technique, we are going to use that to emphasize, to make cells more dispersed from each other, not so crowded together, okay? Just some formulation to describe the mean and the variance. So mean if you treat your variable like a continuous variable, so you do this differentiation with frequency and the small slice of the data, and then multiplicate by the frequency, and then you accumulate from negative infinity to positive infinity. So this is for continuous variable, but you can discrete it using algorithm, whatever. So those are the PDF, mean, and variance table for different distributions. We mentioned Poisson uniform, that's easy, and normal. We mentioned the t-distribution, negative binomial. So those are something we normally discuss in this. I think I also mentioned the gamma distribution. So this gives you a very different shape. If you have alpha and gamma, the two parameter changed, you have a small change in these two parameter values, and you get a very different distribution, whatever you need. So it's very flexible. If you have no idea what your distribution looks like, gamma distribution will fit your data, whatever it is. Flexible shape. Okay, so again, mean, median, mode. Then for this measurement of the disperse, you have a range, deviation, variance, standard deviation. Okay, those are the definition. Also, you want you to remember there's a coefficient of variance, some called the CV. This is the ratio between standard deviation divided by the mean. Okay. All right. So the formula here is to let you to derive an equation like this, which that is if you have a Poisson distributed variable. Based on the definition of the CV is standard deviation divided by mean. Standard deviation is a squared variance. So this is standard deviation by the ratio between these two. Basically, it's a standard deviation normalized by the average. Well, in the Poisson distribution, there's a relationship with a variance the same as a mean. So in that case, we can replace the variance using the mean by itself. So if your distribution is Poisson, then you have a CV definition as a square root of a mean divided by mean. Then you derive this equation here. This is applicable for Poisson distribution. Then you take a log from both sides. Then the log of CV values will be with the slope negative 0.5 log mu. So this creates a linear relationship between CV and the mean. So just remember this. We will use this relationship later to build this curve, this straight line, and you will see in reality, in real data, you found this line here. So can strongly suggest the distribution of UMI is follow the Poisson distribution. And then you will find the genes which jump away from this curve. Those will be used to find outliers in terms of the dispersion level. As I said, it's very important to you to remember how to calculate covariance matrix. But before you calculate the covariance matrix, we need to know how to calculate variance. Okay? So this is your data. You have x1 to x6. You have 17, 15, 20, this column of data. Consider this as an expression of a gene in cell 1, 2, 3, 4, 5, 6. Okay? UMI in gene 1, in cell 1 is 17, this is 15. So how can you calculate the variance of this gene? And you take, so this will be the average, right? So you calculate average first, and then you minus the difference between each your data to its mean. Okay, you get some data which is larger than mean, the 3, some is smaller than -7, smaller than mean. Okay, so your data minus average, so that's what this step is. Your data minus average, then you take a square. You take a square, and then negative values become positive, positive, still positive. So you sum everything together, and you get this number, which is your value minus the average of this value across different samples. Then the difference between each, take a square and then put together. So I just described is here. Then you divide by the total number of samples. You can divide by either n, so 6, or n minus 1. So but statisticians believe n minus 1 gives you a better estimation, so they use n minus 1. But not necessarily. You just divide by n, it will be the same. So this is the variance of this vector of data. So this gene expression cross samples, this variance. When it's large, it means it's more dispersed. So it's unlikely to predict with one single value, because the variance is big. You can either greater or smaller a lot from the mean. So that's what that variance means. Okay? So this value can tell you how variable this gene expression is. So what is covariance? Covariance, when you say covariance, it means two different genes, okay, two vectors. Now, we need to have a pair of variables. So in this case, we have x and y. We will deal with x, y separately first, let x2 minus its mean. So you calculate the average of x, calculate the average of y. So every element in the x variance variable minus its average, you get this difference for the same thing for the y. Okay? Then you multiplicate these two difference. You multiply this with that, this with that. Okay? Then you get the equation of this. It's very similar for the as a variance. It is if you replace y with the same x, then you get exactly the same here, because if you replace y with x, you calculate the variance within the same variable. But now we are calculating the covariance between two variables. So this value, because here is not a squared, not a calculate the square. So if you have positive values for the x here and negative values for the y here, you get a negative product. So at the end, the summation of this could be the negative. In this case, they are negative, so they are these two variables become negative correlated, basically. So this value will tell you how they covariant and also the magnitude. So this is a covariance between two variables. Okay, so I hope you all know this before in other statistic training. So this illustration indicates the way to calculate covariance matrix between 3 variables. So this example is 5 students with 3 subjects. Okay, the score is this one is three columns of data. The way to calculate the covariance matrix, you can calculate pairwise. If you can do variable 1 against 2 using the formula of this to calculate a number, then you can do 1 to 3 and 2 to 3 but the steps here is to using matrix to calculate the covariance matrix between pairwise between 3 variables at the same time, but the same as if you calculate pairwisely. So those formula and to allow you to do the matrix multiplication, and here you basically minus its average. So it looks very fancy, but it's nothing. It's just put the right data in the right position, and when you do the negative, the minus, and then you minor each of this value from its mean of the columns. And then you compute if you do this matrix multiplications, and you get this covariance values. So if you have multiple variables in here, 1, 2, 3, 4, 5, 6 variables, you can plot this scatter plot to look at the relationship between variables. We can see, for instance, between these two, it looks like a negative correlation between this variable versus this variable in this corner. And this scatter plot matrix should be symmetric. This should be the same as this. Some relationship may be positive. So this allows you to have a very quick visualization between multiple variables and see whether their relationship positive or negative related. Translated to our single cell data, this will be gene 1, gene 2, gene 3, gene 4, gene 5, gene 6. And this corner, the scatter plot will be the gene 1 and 2 expression, and each point here is a cell. So you can use this plot to visualize whether these 2 genes are positive correlated. They all expressed when the gene 1 express high, gene 2 also follow in different cells. So covariance matrix and covariance and correlation, they are linked. The formula to translate covariance matrix between X and Y, or 2 variables, covariance to correlation is to use the covariance. So the values here, so covariance, you calculate this. Okay, this divided by the product of the square root of variance X and variance Y is your correlation coefficient. Okay, we use correlation coefficient a lot. Say if you two variables have a correlation coefficient 0.8, that indicate they are positive correlated. Okay. So the compute, or also the direction, is determined by the covariance between these 2 variables. So correlation coefficient basically is a normalized version of covariance between 2 variables. So normalized by what? Normalized by the square root of the variance of 2 variables, the product. This formula makes this covariance matrix variable values is from -1 to +1. You may also encounter the QQ plot. So here I just explain a lot of terminology. You may know or you may not know, but occasionally we will have use this plot to see whether this distribution you observed is the same as your expected normal distribution. In this case, we have normal distribution. Assume all those data points follow that distribution. If that's the case, this QQ plot will be a straight line. And that means your expectation is the same as your observed. What you print here is the quantile values for each observation. So you observe a data in this quantile, say 1%, okay, then your expected should also be here. So this is a test, see whether your expected value observed follow the distribution using QQ plot. That's what the technique we use. If you haven't think about how this will be created, I would say it's harder than usual. You should look at the YouTube and try to understand it. It's a very, very powerful way to show the distribution expectation and see check whether your distributions follow that. So as I said, the last concept we need to have is this KL divergence. So the formula is here. The formula is this divergence is between two distributions. How difference of these two distributions using an equation looks like this. So remember, this is to check whether two distributions. So two distributions, you can use a vector of values in the vector A, and then you have some values. This value may be from the normal distributed data. You generate those vectors. And then you use a B and another vector. So these two vectors represent the samples from two distributions. One is from this red curve, another from the blue curve. So KL divergence will help you to look at these two distributions represented by those samples from A and B, how different they are. You may also heard about another method called Earth move distance. Remember last time we discussed the distance. We haven't got time to mention the Earth move distance. So Earth move distance is also another divergence measurement which described how difference between two distributions is. The idea is if you have a pile of dirt from your construction site from here, you want to move this pile of dirt to there. So change from this to here, how much energy you need to put into move this earth pile from one to that, different shape, different location. That's also a measurement from distribution difference. Let's use a small example to change this continuous variable into histogram and to illustrate how this KL divergence is calculated. So our basic example, this is from a Wikipedia page. So we want to compute the distribution difference between distribution P versus Q. Q is uniform distribution, so you have 0, 1, 2, 3 bins, and then frequency of the same 3.333, 1/3 each chance. Well, this distribution is slightly different. It's higher in the middle, and you have a shape looks like this, and then uniform distribution looks like this. So how can you compute the divergence between these two distributions? So we will compute the difference each bin by each bin. So this will be 1 bin, 2 bin, 3 bin. So this procedure is the same as described here. So this is just a differentiation to slide your data points into infinity number of small bins, but it's the same idea to largely slide your data into 3 bins, same principle. So once we have this, we know the probability for this is 0.3. So that's where the first bin in the Q distribution that have P values, the probability value is 0.03. Well, in this distribution P, the probability is 0.36. So we put the numbers here, so they are probabilities. So these are the PDF functions. Then we move to another bin, bin 1. Then the first distribution have a higher probability, 0.48. Well, the uniform distribution is still 0.033. And the same thing for the last bin. So once we have all these numbers, we calculate the ratio and take a natural log between this ratio. Then multiply it by the first probability. So that's a formula you put in exactly the same here. So you put a PDF values for the first bin here and the values for this ratio between two variable probabilities in that particular bin. So at the end, you calculate KL divergence between P and Q is this. All right. So if you do this same formula, but if you change P and Q different order, then you get different numbers. So the KL divergence is not symmetric. It's different when you do PQ with QP. Okay, sometimes people just calculate both PQ and QP and take average to use it as a KL divergence between two distributions. But I believe this Earth move distance should be the same whether you put your pile of the Earth from one end to another or push back. It should be the same. But we can double check. I didn't put this Earth move distance in the slides here. Questions so far? Okay, everything is fine. We covered quite a lot, I can tell.